Hello there and welcome to this A-level biology revision video with Mr. Phillips, and today we are going to be looking at plant transport and movement of water up the xylem in transpiration. So if we get straight onto this, we need to know is, well, what is transpiration? So you should know that transpiration is the loss of water from the plant via evaporation. So the key word there to remember is evaporation in exam questions, and it's the loss of water from the aerial parts of the plant, so i.e. the leaves, and as you should be aware, the stomata in the leaves. And the reason this happens is that in the mesophyll cells of the plant, of the leaves, the water builds up, it moves into those mesophyll cells, it evaporates, and this then builds up in the air spaces, and then there is diffusion from the inside of the leaf to the outside of the leaf. Now we do call this evaporation, not osmosis, and that is because the water has evaporated, so it is steam that's moving from the high concentration inside the leaf to a low concentration in the atmosphere. So it is the diffusion of water vapor through the stomata of the leaf. So we need to understand this and make sure you can actually explain it. So the transpiration stream happens in the xylem. And what you should be aware is that water will move into the root hair cells via osmosis. It then moves due to differences in water potential from the root hair cells into the root and into the xylem. Now at the same time, we also have minerals such as potassium, let's say, being transported into the root hair cells as well, and that potassium will then, either active transport, lowers the water potential, which allows a water potential gradient to be set up across the cells in the root into the xylem. The water then obviously moves up with the minerals in the xylem vessels, and they move up the xylem to the aerial parts of the plant. So minerals are being carried, and make the note that transpiration is always up the xylem. It never moves down the xylem. And as I said a moment ago, water then builds up in these cells, the palisade cells. The light hits these cells and it causes the water to evaporate. So the water in this area here is steam. You have high concentrations of water in the leaf. You have low concentrations of water outside of the leaf, and therefore water moves from a higher concentration in the leaf to a lower concentration outside the leaf. Because it is water vapor, it is considered diffusion, not active transport. Now, because the water has then evaporated and then diffused out of the leaf, that causes these cells here to have a lower water potential, and then these ones over here have a higher water potential. So water then moves from a high to low water potential via osmosis as a water potential gradient that has set up. And all this happens, all this movement up the plant happens in the xylem tissues, and we'll talk about that in a second. So we need to understand then, well, what is the actual structure of the xylem? So the xylem, as I said a moment ago, is a tissue, because the xylem is made up of lots of cells working together with the function of carrying water and dissolved minerals to the living cells higher up in the plant. And the cells that make up the xylem are called the xylem vessel elements, and they are long thin walls impregnated with lignin. So what you need to be aware of is that the xylem cells are a long tube effectively of plant cells. They have lignin, which is a lot makes them waterproof along the walls of the cells effectively. So they have this waterproof lignin layer. This makes sure that the cells can withstand pressure changes and stops them collapsing in lower pressures when there might be no water available. But also they are waterproof, and hence that lignin is absolutely vital. Now the other adaptation is that the cells are dead when matured. So once they've grown, these walls here will actually decay and die. So what you will end up with is a hollow tube of cells that are all dead, which means that the water can flow up the xylem unimpeded by the walls of the cell. So the adaptations then, just to make it as obvious as we can, is that they are dead cells in a continuous column. They are thin tubes, so the water doesn't break easily. We'll talk about that again in a second. They have pits in the lignified walls that allow movement from one tube to the next adjacent one. And the lignin that is embedded in the cell walls are designed in different patterns which allow the plant to grow in different directions as needed. Now, the flow of water, therefore, how they adapted is not impeded because they don't have end walls, because they're dead, mature cells. The cell contents then are removed. There are no cell contents, and particularly no nucleus, and the lignin in the thickened wall is waterproof, but it also prevents collapse as water pressure changes in the xylem. So again, just to recap about a bit more detail about the water loss in the leaf. So the water obviously arrives from the xylem cells. The water will then naturally move by osmosis from a higher water potential in the xylem where there's lots of water to lower water potential in the cells near and by. Now this is obviously important because this gives the cells water, but at the same time it means then that this is replacing water that is lost. The sunlight hits the leaf, obviously warms the leaf up, obviously more likely to happen from above, I should really say. Okay, that causes the water in the cells to evaporate and become water vapor. Okay, so when the cell's here, it has become water vapor. I've actually covered that up, number one. That water vapor then diffuses from a high water vapor concentration to a low water vapor concentration outside the leaf, and that sets up a diffusion gradient, or so water then moves down the diffusion gradient from an area of high water potential to an area of low water potential where the water is replaced in the. So that's how the water is lost in the leaf, but how does it actually get up the stem? Well, I've got two slides here which will show you this. I actually like the second one more, but I think this is worth showing you. So the first one is, the first theory is, well, part of the theory is root pressure. So root pressure is the idea that the pressure in the roots where there's lots of water coming in is high, and the pressure at the leaves where water is being lost is therefore lower. And we should always use comparative language here. So we should always say higher and lower, because in real life it's not particularly high, but it's higher than it is in the leaf. And because of this higher to lower pressure, there's a higher to lower hydrostatic pressure that forces water into the xylem and up the stem. However, if it was just due to pressure alone, the water would not be able to move up more than a couple of meters. And we know really tall trees can be over 100 meters tall. So there is a lot of pressure. Why do you have that high pressure, as I just mentioned? Because obviously all the minerals have been moved in from the soil due to active transport. Water then follows due to osmosis, and therefore you have a lot of water in the roots as a result. But remember, this root pressure cannot do the whole length of the tree. So how else can this happen? Well, the first thing I want to mention is that there is something called capillary action, okay, or what I would call adhesion forces at work. Now, these adhesion forces are basically the water in the capillary wants to stick to the side of the xylem vessel. So the water is attracted to the side of the xylem vessel, so it's holding on to the side, which prevents it from falling back down as a result. But in addition to that, water is also attached to each other. So because of hydrogen bonds, water has high cohesive forces. And what this means is that one water molecule sticks to another water molecule, which sticks to another water molecule, which sticks to another water molecule, and so on and so on and so on. So these water molecules all stick together, and therefore when one of them gets pulled upwards, that pulls the next one upwards, which pulls the next one upwards, which pulls the next one upwards, which pulls the next one upwards. And that causes a continuous column of water being pulled up the side. And that, along with the adhesion forces that I just mentioned, will mean that the water is sticking to the side of the xylem tube as well, and therefore it's moving upwards. Diagram sort of summarizes that quite nicely. The two forces, there is the cohesion force, which is how the water molecules stick together, and therefore they go, when one gets pulled up, it pulls up the rest of the water molecules, and therefore they're all sort of stuck together as a result being pulled up, like a load of beads on a necklace maybe. The adhesion force then is the fact that the water molecule sticks to the side of the xylem vessel due to hydrogen bonding, and therefore it won't fall back down if the pressure temporarily disappears. So the combined effects of these things can all lead to the water being able to reach the top of the tallest trees. And this really is remarkable if you think about it and you look at the actual height of the tallest trees, are taller than like the Statue of Liberty as a result of this, because they've evolved so they can get the water up there. So there's the cohesion tension theory, which is the idea of the water molecules sticking together. They are a cohesive unit, they stick together. Adhesion forces, which is the idea of the water molecules sticking to the side of the xylem. The root pressure, which is the idea of the high pressure in the roots. Can't write high, sorry, to lower pressure at the top. And the fact that the xylem is very thin as well means that a very small stream of chain of water molecules can go forward. So what evidence do we actually have for this? Well, the main bit of evidence is, well, what happens differently at daytime and nighttime? At nighttime, there is very little transpiration, but at daytime, there is a high transpiration rate. And this is because in the daytime, more water is evaporated from the stomata, and therefore there is a negative pressure in the leaf pulling the water up the xylem. And the xylem, if you actually measure the diameter of the xylem, the diameter or the diameter of the trunk of a tree, the diameter actually decreases in size because the pressure is lower in the xylem in the daytime, because the water is being pulled upwards. Whereas at night time there is no transpiration, and therefore there is less pull on the xylem, so therefore the diameter of the tree increases because the diameter of the xylem. Also, we know that if the xylem breaks and air enters the tree, then we also know that the transpiration stream in that xylem vessel will stop. Okay, so what will happen is this sudden low pressure or unavailability of pressure stops the water moving. So why does the plant do this? Because a lot of people mistakenly think, well, it's just causing water loss. But actually, this is an extremely useful thing for the plant to do. So the plant uses the transpiration stream because it moves water up the xylem. And obviously water is needed to replace the water that is lost. But actually, why is this so important? Well, water is used for lots of key things. So in the leaves, water is used for photosynthesis. It's needed to elongate the plants and grow the cells. And if you think back to meristem cells in cell biology, these are only found in the tips of the shoots and roots. So therefore you need to get to the top of those tips. It keeps the cell turgid. So the cells in the whole of the plant need to get turgid. So water needs to move up the plant to those cells. Minerals need to be carried up the plant as well, particularly things like magnesium that are used in chlorophyll. And evaporation from the leaf keeps plants cool, which allows enzymes not to be denatured in the hottest weather, allowing photosynthesis to continue. So what can affect the rate of transpiration? So transpiration is the rate of water loss from the aerial parts of the plant. And there are several things that can affect the rate of transpiration. So the first thing, hopefully, is the number of leaves. The more leaves a plant has, the higher surface area there is for diffusion, the more stomata there are, and the loss therefore will be greater. The number and size or position of the stomata can be important. So stomata generally are found on the bottom of the leaf as opposed to the top of the leaf to reduce the transpiration rate. Stomata in some plants in drier areas, xerophytes, have less stomata as a result, and they can also have hair around the stomata to reduce water loss. The presence of a waxy cuticle can reduce evaporation, particularly from the upper surface of the leaf, and plants are often like cacti, as in xerophytes, often have this. The amount of light present, and this is the one that is often used as a independent variable in exam questions, because the more light, the stomata will open to do photosynthesis, and therefore more water is lost from evaporation and then diffusion out of the stomata. Another one that can affect the rate of transpiration is temperature, because temperature, with increased temperature, there's increased diffusion rate, and water vapor will move from higher to low concentration quicker, increasing the rate of diffusion. And then the last few, humidity. So humidity is the amount of water in the atmosphere. So if there's a high humidity, that means there's actually a lot of water in the atmosphere. Now what this means is, therefore, you may have, if you had your leaf, okay, and if you've got high humidity, it means you've probably got a high water potential in the atmosphere and a high water potential in the leaf as well. So therefore there is no diffusion gradient between the two. Whereas on a dry day, a non-humid day, there's a high to low diffusion gradient as a result. Now, also air movement and wind is important, because if you've got water collecting around the outside of the leaf, it will stop the diffusion gradient, but with wind, that water's blown away, and therefore it maintains that diffusion gradient. And water availability, if the plant has very little water and extreme drought, it will close the stomata as a result. So how do we actually measure the rate of transpiration practically? Well, what you would use is a piece of equipment called a pedometer. Now, a pedometer is a painful piece of equipment to use in real life, but you need to know theoretically how to use it. Now, the pedometer makes a very key assumption, and that is the amount of water uptaken by the roots equals the amount of water lost via transpiration. Now, in real life, that isn't the case, because obviously the water is used in the plant for reactions such as photosynthesis or for hydrolysis reactions, and therefore it's not 100% accurate, but it is very, very close in terms of measuring it, because so little water is actually used in those reactions. So how this works effectively is you place your plant into a capillary tube, and that capillary tubing has will have Vaseline or some sort of petroleum jelly attached to it to stop any air getting into that gap. You then set this up next to something you can measure a scale with, so a ruler, and you can calculate the diameter of that tubing. And what you can then do is insert an air bubble into the tubing, and then you can record over time, in whatever conditions you decide to change, how far that air bubble moves in a certain amount of time, and that can then be used to calculate the rate, which we'll look at in a moment. But you need to be aware of some of the precautions that you would use to set this up. So the first thing is, you need to ensure that when you actually place the plant into the tubing, you actually do that underwater. So you have a bowl of water and place it in underwater to make sure no unwanted air bubbles get into the apparatus. You would then use petroleum jelly or Vaseline to seal the seals to stop any air getting in. The stem itself, you wouldn't just place a straight stem in. Your stem would have to be cut, not very good picture, sorry. Your stem would have to be cut at an angle, and that increases the surface area to support water uptake into the plant. You would need to control all the other conditions. So often they'll ask you, you're investigating temperature, what conditions should you control? So you should therefore control the humidity, the light intensity, the number of leaves if you're repeating the experiment, the same species of plant, and things like those that you need to think of. And then you would record, as I said, the movement of the air bubble over time to calculate the rate of transpiration. So how would you do this? Because actually you've just got a distance there. What you could use is the volume of a cylinder to calculate the volume of water going through into the plant. So if you use this example down here, the volume of water from wherever it's starting to move. And how you would do that is use this sum here, which is pi r height. Okay, so in this case, pi, let's say 3.14 times the radius squared. Okay, so the radius is half of the diameter. So let's say you had a 1 millimeter diameter, you would be doing pi times 0.5 squared. Okay, and then you times that by the height or the distance moved. So if it traveled 10 centimeters, you would replace that with 10. And that is how you would calculate the rate of transpiration in a pedometer.